Good morning, all. A very warm welcome to everyone for today's Modern Mycology lecture series, lecture number 7. As those who have been joining in the previous lectures, they all know that the Mycoasia and Center for Biodiversity Exploration and Conservation has been organizing this lecture series since past around 6 months, and we have been inviting several speakers from different working in different subject areas and fields. So, and as you all know, that Mycoasia is a journal which is publishing very good research in the recent times, and if we can relate it. So today's what the topic is on slime molds, that is Myxomycota or Myxomycetes, some people called, or the fungal-like organisms. One can, if not the true fungi. So recently, Mycoasia has published an article, very good article on Myxomycetes of sub-Antarctic Macquarine Island by Stephenson. So you can also go through it. So it is a coincidence that in the recent volume, an article on Myxomycota has been published. Center for Biodiversity Exploration Conservation is working on research, doing research on fungi, different aspects of research on fungi, and trying to work on the biodiversity specific aspect, but it's both focusing on basic and applied research. With this, I welcome today's speaker, Professor T.N. Lakhanpal sir. I'll just take a couple of minutes to introduce our today's speaker. Before I start, I think Professor Lakhanpal sir, I do not know whether he remembers me or not, but during my PhD days, on one fine day at the last day of the Indian Science Congress in 2005, I met him personally, because there were since it was the last day of the conference and very less people have turned up for the lecture on time, so we both waited and we discussed on my PhD topic, and I was curious about how to do the sequencing and all those things in the ectomycorrhizal mushrooms on which I was working, and he gave a simple solution at that time, that those which you can clearly identify, differentiate based on morphological features, do not bother to do sequencing. Only those which having the confusing thing and which you are not able to resolve based on the morphological features, you do the sequencing. Since at that time the database was less and still the sequencing was coming up, so he suggested that do not there is no problem. There are still many of the genus which can be resolved by the morphological features, and we had a long talk and I could resolve many of my questions which I was facing. So to introduce sir, Professor T.N. Lakhanpal, he was born in 1944 in a very remote village of Hamirpur district in Himachal Pradesh, and he went to complete his education to Punjab University, Chandigarh, where he also received gold medals in both his graduation and post graduations. He completed his MSc under dissertation under Professor K.S. Thin of Chandigarh and PhD under Professor K.G. Mukherjee in University of Delhi in 1975. And his topic of thesis was taxoecological and experimental studies on Myxomycetes. So it was a pioneering work on cultivation of Myxomycetes and on the study of their life cycle from India. And this work formed the basis of a monograph of Indian Myxomycetes published by J. Kramer in 1982. A very good monograph, a very old monograph, and a very good one. And I do not know, but I hope that is the only one from India. I may be, please correct me later on if I am wrong. He served the university in various capacities and was chairman of the department and also dean of faculty in life sciences. He also served as the director, Institute of Integrated Himalayan Studies, and also visited Department of Forest Science, Oregon State University, USA, Fulbright Fellow, and served in the capacity of professor also. After retirement, he was awarded a Professor Emeritusship by UGC. He also served for a brief period as a professor in Mizoram University and also as a visiting professor in Department of Bioscience, Sri Satya Sai University, Prasanthi Nilayam. Dr. Lakhanpal has a pioneering research work on cellular slime molds, acellular slime molds, mushrooms, and mycorrhiza. Besides this monumental work, he has also done very good work on the biology of the Morchella. and also developed the technology of cultivation for shiitake, that is Lantana acidotis, and milky mushroom, that is Psilocybe indica. Dr. Lakhanpal sir has been honored with Best Teacher Award by Himachal Pradesh University, Shimla, Saraswati Award by Delhi University, and also he served as a president of various societies, including the Indian Science Congress of Plant Science Section, Mycological Society of India, and also Indian Mushroom Growers Association. Sir has a credit of more than 190 research papers, 55 chapters, more than 6 monographs, 9 books, several edited books, and also trained MSc and MPhil students, and 30 students have received PhD under his supervision. This I welcome Dr. Lakhanpal sir. So very thank you for taking time out and giving the opportunity all our participants to listen to you on Myxomycota. Welcome, sir. Welcome. Uh, I yes, sir. Yes. Sir, will you able to present the presentation or share? I move the slides from here. I can
No,
help you.
you please, you do it.
Okay, fine, sir, fine, sure,
You do it.
sure,
I will simply keep on explaining.
sure, sir, sure.
Uh, So, on the outset, let me first thank you. And I remember, I remember your earlier face, not the new one,
Yes, yes.
because you, as I have changed, you have also changed, and constantly you have been in touch. That is a good thing, and you have been constantly doing something or the other good research work on the subject. So I am grateful to you for giving me this opportunity to interact with, I see many people, some of whom I know very well. And thank you very much for, I see Dr. Pradeep from Kerala. And now, thank you very much for the kind introduction. Now, what I do is, because they all are very experienced people, but I do not know how much familiar most of them are with the myxomycetes or slime molds. And I started my career with slime molds, then diversified. And I am happy that some of our discussion that was fruitful and that could help you in taking forward the problem that you were facing at that time. Now, what I will do is, there are a few terms which would be frequently used in the lecture. So, one of the terms is mixed amoebae and swarm cells. Mixed amoebae are amoeba-like structures which the spores, which ooze out from spores in myxomycetes. When they are amoeboid, we call them mixed amoebae, and when they have flagella, we call them swarm cells. So these would be frequently used. Another term which is important and very significant, that is free nuclear division. Now, myxomycetes are unique in that their nuclei divide without cell wall formation, and so it is called free nuclear division. Another characteristic feature is synchronous. The term is synchronous nuclear division. Synchronous because all the nuclei in myxomycete plasmodium, they divide simultaneously, and if we fix, they are at the same divisional state. So that is what is meant by synchronous nuclear division, simultaneous. Another important feature is protoplasmic streaming. The protoplasm in myxomycetes shows a very rapid, brisk movement, and that is what is a very significant feature in slime molds, especially in these true slime molds, and that is what is meant by protoplasmic streaming. Then, with regard to the uh, fructification development, we normally use two terms: subhypothelic and epihypothelic. Hypothallus is a structure which is present at the base of a fruiting body. So some grow on that, that is epihypothelic, and there are others which grow below that, and hypothallus is laid down later. So they are subhypothelic. So these are some of the terms which we will frequently encounter, and I thought let me first clarify them.
Yeah. Sir,
Now,
I hope you will instruct me to change the slide whenever it is required, sir.
yes, yes. Okay. Oh, I will keep on. I will say yes. I will say tell that, sir. So, myxomycetes, they have been placed differently by different workers, and in the present system of classification, they are no longer considered plants. But nevertheless, this is a group which has been studied and is still being studied by mycologists. So that is one of the reasons why they are included in mycology. But myxomycetes, they are better called as Switzerland of biological kingdom. They are Switzerland of biological kingdom because they do not belong to any specific groupings. They lie independently, and therefore they are friendly with all. They have no enmity with anyone, so that is why they are like Switzerland. They are also very curious because curious in the sense that they are on the borderline of plants and animals. They do not follow the traditional biological classification system. They have characteristics of both plants and animals. Half of their life cycle is plant-like, and the other half of their animal's life cycle is animal-like. So the animal-like phase, which is called assimilative phase, because most of the metabolic activity takes place in this is called plasmodium. They this plasmodium is animal-like. The previous one, please. So the this is the animal-like phase, and this animal-like phase then transforms into a plant-like phase with spores born in sporangia. So, and they have a spore wall, so that is plant-like. Where they show a mode of nutrition which is saprozoic like plants and holozoic like animals. Next, please. Now, this is an outline cycle, and the spores germinate when there are suitable conditions of moisture and temperature and proper pH, almost neutral pH. And the germination is by two methods. The spore wall, when it swells up, then either it cracks or enzymatically there is a hole formation, and that hole or pore, when it is formed, or a crack which is wedge shaped when it is formed, it releases the protoplasm outside, and this protoplasm which is released from the spore, this has a amoeboid shape, so it is called a mixed amoeba. And the mixed amoebal phase, or the mixed amoeba, sometimes or in times depending upon the species, it develops flagella which are heterocont. One is smaller and other is the larger. Then it is called a swarm cell. It lies on the anterior end, and the posterior ends are sticky in mixed amoebae also and in swarm cells also. Now, mixed amoebae and swarm cells both, they divide and undergo multi-divisional stages and form a large population either of mixed amoebae or of swarm cells. The same swarm cells or the same mixed amoebae, then they fuse in pairs. That is the beginning of sexual reproduction. They behave as gametes. And the fusion is at the sticky posterior ends. After fusion, then there is the formation of a diploid zygote. They are all haploid. Spore is haploid. Mixed amoeba and swarm cells, they are also haploid. But the zygote becomes diploid. But zygote does not develop a wall. It is a naked mass of protoplasm. And this diploid zygote, the diploid nucleus in a zygote, it divides simultaneously and synchronously. So there is a free nuclear division. It becomes a multinucleate structure. And in this multinucleate structure, all nuclei are diploid. Now, this is called plasmodium. It is a mass of protoplasm. Zygotes may also be formed by the coalescence of different zygotes. Many zygotes may fuse. But the interesting point is, whenever they fuse, then the nuclear division, within 3 to 4 hours, this nuclear division becomes synchronous. They develop synchrony, and all the nuclei, they start dividing simultaneously. So that is what is meant by there is no cell wall formation, so it is free nuclear division. There is uh, and the nuclei are at the same divisional stage, so it is a synchronous nuclear division. So this mass of protoplasm then undergoes various morphological changes. In some groups, it is inconspicuous in the beginning when it is called protoplasmodium. In some, it is inconspicuous, becomes conspicuous later, then it is called aphanoplasmodium. In others, it is conspicuous or visible right from the beginning. The conspicuousness or inconspicuousness is because of the microscopic examination. They cannot be seen with the naked eye. They, however, can be observed under a stereo binocular or low power of a microscope. So, if they are visible right from the beginning, then it is phaneroplasmodium. If they if we just see a speck and then it enlarges and forms a protoplasmodium, then it is a phenoplasmodium. But if it remains microscopic and is not easily visible to the naked eye, even under the microscope, then it is a protoplasmodium. Protoplasmodium ultimately gives rise to just one or maximum two fruiting bodies. Phenoplasmodium produces slightly larger number of fruiting bodies, whereas Phaneroplasmodium produces a mass of sporangia. Now, the conspicuous feature is that a Phaneroplasmodium, the diagram which is net-like, if Phaneroplasmodium, there is a differentiation of the plasmodium into veins and veinlets. And there is the thick structure that you see in this diagram in the plasmodium is that is the anterior end. and this anterior end is thick where the protoplasmic concentrated, and these veins and veinlets are the streams which emerge from this thickened portion of the plasmodium or the protoplasm. If we just focus under the low power of the microscope one vein of the plasmodium, then because it has debris, spores, it is holozoic as well as saprozoic in nutrition, so small particles can be seen, and there is a brisk movement of the protoplasm, and this movement is very characteristic movement, and this characteristic name is given because of the fact that if you focus it under the microscope, the stream of protoplasm will move in one direction. It moves in that direction for 20 to 25 or 35 seconds. Then it becomes stationary for 5 to 10 seconds. And then it is reversed. It will start moving in the reverse direction. So it is very fascinating to examine the protoplasmic movement or observe the protoplasmic movement. And since because it forms a rhythm of forward movement and then a backward movement for and a intermediate stationary phase, so that is why it is called rhythmical reversible streaming of the cytoplasm. So this is a very characteristic feature of a phaneroplasmodium. This feature, and what is the cause of this movement? It was reported in the 1950s, and protozoan actin and actomycin and myosin fibrils. It is reported that when they interact, when there is a change in pH, so actin fibrils and myosin fibrils they interact and they result in the movement of the cytoplasm. This movement is prominent in case of a phaneroplasmodium, is feeble in an aphanoplasmodium, and is very insignificant or not at all present in a protoplasmodium. This was something which was first discovered in 1961 by Alexopoulos, and before that we simply called them as plasmodium. The classification and this plasmodial types, they have a bearing on the classification of the myxomycetes. So these have now been incorporated in the classification system, and they have a important role and they are characteristic. There are some variations also, but nevertheless these are the three prominent types which are met within this. Now this plasmodium, when it reaches a certain stage of maturity, and there are various factors which determine. Light is one, temperature is another. If it is disturbed, the disturbance to the plasmodium, or if you prick it with a pin, then it again resorts to vegetative phase. So fruiting occurs at a specific time, and during fruiting, the condensation of the plasmodium specific of a species that takes place, and then the fruiting body is molded in the manner that is genetically characteristic of a species, and then spores are delimited. Rest this cytoplasmic mass which is left unutilized, that comes down and settles at the base, or that is what is called as hypothallus. These fruiting bodies which have been shown, and this structure which is shown at the base is the hypothallus. And then the spores are differentiated. The meiosis takes place here, and after meiotic division, the spores are delimited, and then the cycle is repeated. So this is in a nutshell, briefly, the life cycle of a slime mold. Next, please. Now, They are not of any direct economic importance, but they are associated with decaying vegetable matter. So that is why we should study them. They, the plasmodium is the richest and most massive source of pure form of protoplasm. And uh, if we know that they have been, it has been an ideal material for study by biochemists, physiologists on fundamental aspects of cell metabolism, and whatever 90% of the knowledge that we have on the characteristic features of protoplasm or characteristics of protoplasm, that is derived from the plasmodium of myxomycetes. So they, because it is the purest form of protoplasm, many species have been used for producing enzymes and protein biomass. Now, another important aspect has been that since in cytoplasm the division is synchronous, the division is free nuclear. Free nuclear division occurs in cancer cells. So in since from in MIT, most of the cancer research that has been done on myxomycetes, and there is one species in particular which has been used, that is Physarum polycephalum, mass produced, because they still not much headway has been made, but nevertheless they want to know, in case of myxomycetes, when the nuclei divide, there is free nuclear division, but then they resort to metamorphosis into fruiting bodies, and it is a controlled nuclear division. Whereas in cancerous cells, it is a uncontrolled nuclear division. So that is why the cancerous cells, they keep on dividing. So they just want to isolate the element which initiates and stops the nuclear division in a myxomycete plasmodium, but not in cancer cells. Maybe if there is some success, then maybe some element is isolated which can be utilized for restricting the growth of cancerous cells. Now, this is a large, plasmodium is a large single cell, so it is considered a single cell, and just as it is given in the last sentence, exhibits remarkably precise mitotic synchroning and breaks to and fro cytoplasmic streaming of the cytoplasm. So, not exactly that they are of direct economic importance, indirectly, since they are associated with vegetable matter, they are purest form of protoplasm, they have peculiar characteristics which may have answer for some of the questions which are posed in cancerous cells. So that is why we need to study them. Next, please. Taxonomically, we have recorded around 500 species. The world over, there are around 2000 species. And the taxonomic criteria which are used, they are types of plasmodia. I told you protoplasmodium, aphanoplasmodium, and phaneroplasmodium. Then types of fruiting bodies and the fruiting body components. Next, please. We will come to them one by one. Next, please. Can we go to the next?
Rohit, are you there?
Next, please.
Rohit?
Yeah. These are the plasmodial types: protoplasmodium, aphanoplasmodium, and phaneroplasmodium. I have already detailed them. Next, please. This is The first one is the protoplasmodium. The second one is the phenoplasmodium, and the third one is the phaneroplasmodium, and fourth, the enlarged one is also phaneroplasmodium. In the protoplasmodium, you can see this is with the microscope, a small, there is a small membrane which surrounds it, but it is not visible in the beginning and remains inconspicuous even at later stages. A phenoplasmodium is conspicuous in the beginning, inconspicuous in the beginning. Later on, it develops veins and veinlets, but they are not very conspicuous, and it lacks rhythmical, both of these, proto, pheno, they lack rhythmical reversible streaming of the cytoplasm, whereas in phaneroplasmodium, you can see the veins and veinlets and the expanding fan, and this is how it looks in nature, and they are variously colored. They are very brightly colored plasmodia as well as the fruiting bodies, and these are these veins which exhibit the synchronous this rhythmical reversible streaming of the cytoplasm. Next, please. The fruiting types, mainly there are these three: plasmodiocarps, ethelia, and sporangia. Next, please. One by one, we can see them. Plasmodiocarps are attached to the substratum. They are pressed to the substratum. So there are their individual. So this first one is the plasmodiocarp. They may be longer than broad, or they may be isodiametric, or they may be just flattened, but they are attached to the substratum. They do not have a stalk, and the hypothallus is suppressed, or they are immersed in the hypothallus. Or plasmodiocarps may form a network as is present in the first photograph on the right side. This is Hemitrichia serpula. The plasmodiocarps may be joined together to form a reticulum, and this is called network of, or this also is the plasmodium carp. The fruiting bodies, they are spherical or elongated, sessile, or with the stalk. And the second one on the right side is a pseudoethelium. This is called ethelium. This is called sporangium. Sporangium is globular in structure, has maybe sessile or has a stalk. Many sporangia, when they get fused together, or many plasmodiocarps, when they fuse together, they form a pseudoethelium. This is called ethelium, is a congregation or a massive fruiting body. And in most of them, why do we say them pseudoethelia or not true ethelia? Because when we dissect them, we can see the partitioning of the walls, but the walls do not extend from periphery to the center. So therefore, it can be assumed that they have not been partitioned completely, and incomplete partition because of the clubbing together of the fruiting bodies, they are called pseudoethelia. The, this small sporangia you can see, and this is a typical sporangium of Stimonitis. We have a stalk, and then there is a sporangium. Stalk and sporangium, so they are plasmodiocarps, intricately networked plasmodiocarps, ethelia when they are single and without any partitions, or pseudoethelia when there are partitions present inside, and the sporangia in mass, and these are stalked sporangia. So sessile sporangia and stalked sporangia. So these are the different types of fruiting bodies which are met within the myxomycetes. Next, please. The fruiting bodies, they have these components. Hypothallus is the leftover cytoplasm deposited at the base. Stalk, all of us understand, is the stipe or stalk which holds the sporangium or supports the sporangium. Peridium is the wall which of the sporangium which encloses the spore mass. Inside the spore mass, sometimes there may be an extension of the stalk. When stalk extends into the sporangium, then it is called a columella. Sometimes the columella is not an extension of the stalk, but it is simply some extraneous matter which has been deposited, but is prominent and discernible. So that is called pseudocolumella. And then there is a thread-like network which they form, that is called capillicium. Capillicium helps in spore dispersal. It is elastic in nature, and pseudocapillicium is when it is not formed from the stalk, but either it intrudes from the peridium or it develops from the spore mass, and then they are disposed. So hypothallus, stalk, peridium, columella, capillicium, and spores, these are the different components of the fruiting bodies. Next, please. And this shows the fruiting bodies, stalk fruiting bodies, sessile fruiting bodies, intricate fruiting bodies or network of the plasmodies, and these are all stalked fruiting bodies. Next, please. This is the columella when the stalk extends into the sporangium, and pseudocolumella when the stalk is not an extension of the columella, then it is a pseudocolumella. Next, please. These are the different types of capillicial threads. It may be just an amorphous mass, or it may be a tubular mass of tubes when it is tubular, or it may form a network and intricate network. It may be like elaters, just as we have in bryophytes. So elater-like or network or tubular structures, or they may have some specific markings on them of different types. And these serve as good basis. The plasmodial types, the fruiting body types, and the various components and the capillicial types, they all serve as good criteria for delimiting different species because they are characteristic of different species and therefore easily discernible and can be easily utilized for distinguishing different types of species. Next, please. These are the photographs. So you can see the network, you can see the cog-like or ring-like structures, you can see the spine-like structures. You can also see this network. This is a beautiful network that they form, which may be open or which may be closed, and this holds the spores. And since it is elastic in nature, so it helps in the dispersal of the spores. Next, please. These are the different types of spores. Spores bear different types of markings. They may be smooth, they may be warted when they are spine-like, they may be joined together, or they may form a network, or they may form incomplete reticulum or a complete reticulum. So again, these are very good criteria for distinguishing different species or within species, interspecific and intraspecific. These are the different types of stalks. So stalk may be hollow, stalk may be stuffed, or stalk may be swollen and with granules, or it may have the fibrillar material, or it may have spore-like cells. In fact, these are the sterile spores which have not metamorphosed into normal spores, so they get deposited in the stalk and rest of the cytoplasm that oozes out and is deposited at the base of this. Then another characteristic feature, especially in the higher members of myxomycetes is the presence of calcium carbonate crystals or globules, and either they are spherical or they are sharp or they are of different shapes. And these different types of calcium depositions, they have a marked influence on the classification of different genera or different species and even in the orders. Next, please. So this shows. Now this is Dictydium, this is Arcelia, this is Diderma, and this is Lepidoderma tegrinum. You can see tiger-like skin specks on the wall. So peridium also has various shapes. The peridium here is blown off, that is evanescent, that is transitory. It falls off and leaves behind this network. And in this network would be entrapped the spores. This is Arcelia. Arcelia, the, this is the network, and the network expands on discience because it is elastic in nature, as I told earlier. So this elasticity gives them the advantage for spore dispersal, and this all is capillation. This is a columella extension of this type, and this is peridium. Diderma has double peridium, so diderma, and lepidoderma because it looks like the skin of the loin. So the various names, as is true of other groups also, various names earlier were on the likening. How do they resemble the different parts? And that is how they were initially explained. And we still continue with them because they have valid and meanings and in the delimitation of the species. Next, please. Now, this is with regard to the sporophore development. Now, in the development, as I said earlier, there are two types, major types of development: subhypothelic and epihypothelic type of development. Now, on the left side is the subhypothelic development, and on the right side is the epihypothelic type of development. Though it is not very clear, but the cytoplasm condenses. In the first slide you can see, first projection, first photograph, you can see this is the condensed photograph, cytoplasm. This condensed cytoplasm is forming the spherical bodies, and these are the sporangial initials. These sporangial initials start elongating, and hypothalamus is deposited first. इसमें चला गया है the देखने मिला just a minute, wait for a second, ये साइड पे चला गया इसको बना हो जाएगा कौन सी स्लाइड यही है it is the same, so let it has come. Double tick. So these sporangia are elongating, hypothalamus is deposited at the base, so that is why it is called subhypothalic. And then these are the mature sporangia, stalk and hypothalamus at the base. You can simply guess that it is there because it is always there. This is in normal. This is the Myxogastromystidae, one class. This is Stemonitomystidae, the other class. In Stemonitus-like species, the sporangia initials are formed first. Sporangia develop, stalk develops, then the sporangia mature again, and at the base, and at the base is deposited the hypothalamus. So it is subhypothelic and it is epihypothelic. So the deposition of the hypothalamus and then the formation of the sporangia, that is important. Epihypothelic type of development is found in stimon. This was the work of Ross, and that was in the 70s. And then initially we treated myxomycetes as one group, but on the basis of the plasmodial types, Plasmodium in stimonitis is a phenoplasmodium, and the development of the fruiting body is epihypothelic. So, on the basis of these two major characteristics, this was segregated as a Stimonitomystidae and Myxogastromystidae, and another is Serratiomyxomystidae, that is called exospory and endospory, because in Serratiomyxa the spores are borne externally on stalks. In Myxogastromystidae the spores are borne internally, endospory, and now we have these three subclasses. Next, please. This is how it is depicted. So the hypothalamus is deposited, sporangial initials are initiated, the cytoplasm moves into the sporangial initials, the sporangial initial enlarges, it takes up or transforms into the specific shape of a fruiting body. It is destined to be genetically. So the cytoplasm movement, then stalk is formed wherever the stalk is present. In sessile forms, no stalk is formed, and then this spore cytoplasmic mass with multinuclei, then it is delimited into uninucleate pieces. So uninucleate spores are formed after meiosis, stalk is laid down, and then at the base hypothalamus is already formed. In the second type, the sporangial initials begin earlier, they grow into sporangia, form the sporangia. This is the columella, the stalk and the columella. The extension into the sporangium is the extension of this type. So this is columella, true columella. And then whatever is left after utilization or formation of capillicium and spores, that is deposited at the base. So that forms the base or the hypothalamus. So it is subhypothelic and it is epihypothelic. Next, please. So this is what I said. On the basis of the types of plasmodia and on the basis of the type of sporophore development, we have now three subclasses: Serratomyxomystidae, Myxogastromystidae, and Stimonitomystidae. Serratomyxomystidae is called traditionally exospory, and these two are called endospory. Serratomyxomystidae has spores which are borne on stalks externally. Myxogastromystidae has a peridium which encloses the spore mass in a sporangium, and the Myxogastromystidae has a phaneroplasmodium and a protoplasmodium, and Stimonitomystidae has exclusively a phenoplasmodium and a epihypothelic type of sporophore development. Next, please. This is Serratomyxomystidae, Serratomyxa. So in Serratomyxa, the spores are borne externally. We can see them under the microscope. The spores are visible. They are glistening white, and Serratomyxa is the first, which comes first during the rainy season on decorticated bark or the felled trees or stumps. Next, please. This uh, this is Trichia phagogenia. You can see the stalk. You can see the sporangium. So sporangiate fruiting bodies. Next, please. They're beautiful colored. This is yeah. You can see this. These white specks are the calcareous structures of capillicium. This is in didymium. The This is the capillicium, and capillicium is thread-like, and these threads are formed of the capillicial mass. So capillicium shapes itself into structures which give a definite shape to the peridium, and then it encloses the spore mass. Next, please. This is arcelia. I shown earlier also. The arcelia, the fruiting bodies, elastic capillicium, and then the hypothallus at the base. Next, please. This is stimonitis, hypothallus at the base, formed later, sporangia. And there is a very interesting story. If those who have not read may read the introductory part of the book by Alexopoulos, the introductory mycology. There he gives that there was an exhibit in an exhibition, and stimonitis fruiting bodies were placed there, and it was written, the caption was hair growing on wood. And Alexopoulos writes that all believed that it is a hair growing on wood, but mycologists did not. Next, please. These are plasmodiocarps, sporangiate plasmodiocarps, and they are sessile without any stalk, and the hypothallus is at the base. It can be seen with the microscope. They, you know, they are not parasitic. It is shown simply on a, they can grow on living leaves or living twigs. They are not parasitic, but when the plasmodium is growing and when it crawls, wherever it crawls, it looks for a dry place. If you grow them in a petri dish and keep on watering, when the mix amoeba or the swarm cells are formed or plasmodium is formed, if you keep on pricking it, if you keep on uh, providing it with water, free water, then invariably they will crawl to the upper lid and fruit on the underside of the upper lid. So that shows that they look for a drier place. And we do not know what exactly that is, but they look for a drier place. That is number one. Second is that there is a time when they reach a point of no return and they must fruit. So then they fruit. So uh, a number of factors have been proposed, that it is because of light or because of pH, or many explanations have been given. But plasmodium ultimately reaches a point of no return, and then it must produce the fruiting bodies. Next, please. So we can skip because these are different monographs. They will not interest. Next, please. This right side, this photo I had included simply because he is the person who revolutionized the taxonomy of slime molds. There are three types of slime molds: cellular slime molds, acellular slime molds, net slime molds, and dictyostelium or acellular slime molds. They He found the link. If you look at the 62 edition of Felixopolis, there is a group which is called Order Acrasiales, an order of uncertain affinity. So this person he discovered groups which link the Acrasiales to Myxomycetes on one hand, and on the other hand to protozoan members. And there is a large, he worked in 69 and for almost 20 years, and he and his students, they have published a lot of work on these slime molds, which are linkages between the different groups. Olive. His name is L.S. Olive. Next, please. C.J. Alexopoulos and L.S. Olive. And C.J. Alexopoulos was my PhD examiner. Next, please. Next, please. No, the same previous one. The previous one, Rohit. Alexopoulos, Olive, and this is Covening. Covening, this photograph I included purposely because Covening's thesis is not a written thesis. He was the first person who time lapse photography, because myxomycetes they fruit at night, they are nocturnal. So he had devised and put up a photomicrographic attachment which could automatically photograph them, and that is how he submitted his thesis in Iowa State University with Dr. Martin, who was teacher of Alexopoulos, who he submitted that filmed life cycle for the first time of the myxomycetes with time lapse photography and all the stages from spore germination to these swarm cell formation to mix amoeba formation, fusion into zygote, development into a plasmodium, formation of a plasmodium, and then development of fruiting bodies. They were all photographed by Covening. Next, please. This is a monumental monograph. There are many, starting with 1895 by Lister, 1911 by Lister and Lister, his daughter and he, 1925. Then it was treated as mycetozoa. They were included in protozoa. The myxomycete term, there are two schools of thought. The British school of thought, they considered them mycetozoa and treated them along with the protozoa. And American school, they consider them plants, because there is a beautiful article by which all should read. Even now it is valid. This was in Botanical Review, written by G.W. Martin, University of Iowa. Are fungi plants? It's a beautiful article, a review article, and very convincing logical article. And so this was a monograph on world myxomycetes by G.W. Martin and C.J. Alexopoulos. Next, please. Then they revised the genre of myxomycetes. Mary L. Farr, she authored this. Next, please. Now, this is Professor K.S. Thin, Dr. Agni Hetrudu and Indra Kalyan Sundaram from University of Madras, and Professor K.G. Mukherjee from University of Delhi, who both of these have been my teachers, and they have worked on myxomycetes of South India. They have worked on myxomycetes of North India and Northeast India. Next, please. Just as Rohit was saying, the first monograph on Indian myxomycetes was in unpartitioned, undivided India, Indian slime molds by Sultan Ali Lodhi from Punjab University, Lahore. It had around 35 species described. Then Myxomycetes of India was published by K.S. Thin in 1977. We published Taxonomy of Indian Myxomycetes, and which was published, as Dr. Rohit mentioned, by Kramer. And now recently in 2024, Dr. Rajesh Sharma and myself, we have published a monograph on myxomycetes of Northeast India and Royal Kingdom of Bhutan, and that has been published by uh, Bishan Singh Mahendrapal Singh, because rights of Kramer, they were purchased by Bishan Singh and Mahendrapal Singh. So even earlier monographs they are available with Bishan Singh, Mahendrapal Singh. So that is the first ever treatment on the myxomycetes of Northeast India. Next, please. So this is Dr. Thin's monograph, and this is our monograph. Next, please. So I don't think this will interest people, collection and care, and how they are to be collected in nature and collection. Many species they have been isolated from moist chambers on bark, which are microscopic, and how they are to be studied. These are of practical application. Next, please. And this is how the what do we require and how the specimens they are revived and Aman's lactophenol is the mounting medium for species which do not have calcium. For species which have calcium, the calcium dissolves in lactophenol or lactic acid. So there we use only glycerine. Next, please. Um, these Stephenson, whose name was mentioned, Stephenson was with me. We worked together here on myxomycetes, and he was previously in. Now he is in Arizona State University or some other university, but previously he was in a college near New York. Next, please. So myxomycetes from bark, from litter, from dung, from soil. They some are specific, some are very versatile. They are habitat relationships. Next, please. And moist chamber by through this moist chamber technique, microscopic forms can be isolated, and we have done a lot of work on the corticolus myxomycetes. Maybe in future we process it, and then we because 85 species out of which 45 are new records and 21 are new species. Maybe we process it into this monograph. Next, please. Then they grow on different substrates, and they are some are substrate specific. These give the names of the people who have done work on coprophilus or corticolus myxomycetes. Corticolus myxomycetes, the first report was in 1933 by Gilbert and Martin. Gilbert was teacher of Martin, G.W. Martin, and Martin was teacher of Alexopoulos. So there is a pedigree of people who have devoted their life, and there is a lady, was a lady naming Abramica who was a mature. She has done lot of work on myxomycetes of Netherlands and published lot of work. She, in fact, was a good authority on myxomycetes. Next, please. Ecological studies. Um, Most of the ecological studies have been done by Stephenson. Stephenson is basically an ecologist, and he has worked. Most of the genetical studies, it is a very good material for genetics and biochemical studies. Colin and his group at California State University and Gray, Parr, all these people, they have worked on different aspects of myxomycetes. Next, please. Next, please. This different substrates. This only gives the substrates. This gives what we have done. So I told you it is two species and three varieties, isolated and next, please. This gives the list. The list with now the northeastern. Next, please. The total number of species is around 400. Next, please. This gives the pH range. there are no pathogens in myxomycetes. They simply crawl on the substrate, but they are ideal or model organisms for studies in biochemistry and physiology. They have novel chemical compounds, and even there are uh, not Stephenson exactly, but there are others who are working in US on different aspects of Keller is one who has worked on Keller and Kolsky. They have worked on the corticolis myxomycetes. Next, please. And this still holds good, though it was Hagelstein stated in 1944. The collection and study of myxomycetes present an almost untrodden field. There have been few studies and there are few today as compared with the many in other pursuits. There are opportunities for research not only in morphology and physiology of the group, but in taxonomy. And from a biochemist's view, slime molds offer similar advantage as bacteria. Such an organism can be grown even in larger for matters its organelles and nuclei and mitochondria, and even nuclei can be isolated and thus studied separately, and no rigid wall surrounds the plasmodium. Its macromolecules can be extracted relatively easy. This was stated in 1933, and just as dictyostelium discoideum has been used as a in acellular slime molds as a model organism, in a similar manner, Physarum polycephalum is one which is being used, and there are many others because we worked on acellular slime molds. Um, I had a fellow from US, Athens, who Cavender. We worked on the acellular slime molds, and we just recorded 12 species from sea level to the Himalayan ranges. But myxomycetes, we have recorded around more than 500 species. So there are a large number of species. They can be cultured, and the field is open, and sky is the limit. Next, please. So systematics, laboratory cultivation, then its utilization in different manners that can be exploited, left to the myxomycete research for future research opportunities. Next, please. So thank you very much. Thank you very much for your patient listening, and thank you very much, Dr. Rohit, for giving me this opportunity to interact with colleagues from different parts of the country. Thank you very much.
Thank you so much, sir. Thank you very much for the very nice talk. Uh, I think those people who have questions, please raise your hands. And by the time people are thinking about their questions, sir, I have a small question that about culture because a few days ago, Mr. Rain, he's also joined the talk. He's online from Raigad district near Konkan region of Maharashtra. He has collected some stemonitis on leaves, and he asked me how I can culture them. So in the moist chamber, how one can proceed to culture
No,
the.
simply what he has to do is take a cavity slide, put a drop of water, not distilled water. You can put rainwater or tap water because they feed on bacteria.
Okay.
They feed on fungal spores. So that will hasten the germination. So in the cavity slide, you can put a cover slip and just a drop and put a few spores, because large number of spores, they become competitive. So if you can put just 2 or 3 spores, then he should go through the paper, because Indira Kalyan Sundaram worked on Stimonitis herbatica, on the culturing of Stimonitis herbatica. He may get some clue from there. Otherwise, a lot of literature is there. I don't know which literature he has approached to, but they germinate just like any other spore. Putting a cover slip is, if you can focus it under the low power of the microscope, and it is a fascinating phenomenon to watch them. If it germinates, then swarm cells or mixed amoeba will come out, and then they start moving about.
Okay.
So
Okay.
once that happens, then he can simply transfer it to low nutrient agar medium,
Okay. Okay. Okay.
and if possible put a bacterial culture alongside,
Okay. Okay.
or tap water.
And low nutrient agar medium will be PDA, MA, this kind of media only, sir?
Yes, the
Okay.
same.
Fine. Thank you, sir. Thank you so much.
Welcome.
Next, Sheetal Chaudhary, please go ahead with your question.
Uh, Good afternoon, sir. First, I would like to thank you to Mycoasia also and to you also for bringing such a good topic. Very ignorant, I think, because most people do not care to study slime molds now. So I'm very happy that I got the opportunity to learn it from you. So my question is, sir. During the life cycle, the plasmodia, if it doesn't get the proper conditions for fruiting, sometimes it undergoes certain stages, resistant stages. We call it sclerotia.
Yeah,
Uh,
they form sclerosia. Yes.
yeah, so in that case, it is very difficult to identify morphologically the species. So, can you throw some light over it? How we can identify if
No,
it
it cannot be identified at sclerotial stage. For
okay.
identification, only fruiting bodies are essential.
Okay,
So
but
what you
can we
can do
count it?
is, the sclerotia, if you put, if you moisten them,
Hmm.
they will re-germinate.
Yeah, okay.
And then, feed them properly with bacterial culture or oatmeal.
Oatmeal.
Oatmeal, if you put, then sometimes what happens is the oatmeal grows on the surface, collects on the surface, and then it shadows the fruiting bodies. And
Hmm.
oatmeal also catches aspergillus or penicillium as they grow on it. So that can obliterate the vision. But if you put some bacterial culture, E.
Hmm.
coli is the best. It feeds on E. coli very well. Or take an extract of oatmeal and then put it there. The sclerotia regerminate, and then allow it to gradually, when if you are growing them in a petri dish, and if it
Hmm.
crawls, uh, if there is enough moisture, then it will crop to the walls and even to the lid.
Okay. Hello.
So because they are nocturnal uh,
Hmm. Hmm.
in fruiting body, so you have to put them in the dark.
Okay.
Don't put them in the light. If you put them in the light, they will vegetatively keep on growing.
Right. Okay. Thank you, sir. Thank
Thank you,
you.
Sheetal. Thank you, Sheetal. Srijit, please go ahead with your question.
Good afternoon, sir.
Good afternoon.
It was a really interesting and informative talk. Thank you for that.
Mm,
And I am from Kerala Forest Research Institute.
mm,
Actually, recently we have started working on myxomycetes, and we have collected samples and identified almost 40 or 50 species. And
okay.
we are in the process of establishing a collection slimarium in our lab. And my first question is that uh, where we have the largest collection of myxomycetes or the slimarium in India, or we have such a whether we have such a reference library
For
in
no,
India.
unfortunately, the Best collection was at Punjab University, Chandigarh,
Hmm.
and then US in botany in Madras University. I don't know.
No,
No, who is?
Madras University, the collection has lost due to some flood or something like that. I
Yes.
heard it.
So uh, it has been lost because of flood in Chennai and
Uh-huh.
because of human flood in Chandigarh.
Both are lost.
Yeah. So you won't find many. But if you need any help,
Uh-huh.
you can just you can come to me. We can do that, or send the specimens. I can identify.
Thank you very much. Sure, sir.
No,
Sure.
no problem.
Sure, sir.
And
Thank you very much. And one more question, based on my little knowledge, these
no,
type of organism are very widely distributed. For example, we don't have that much of endemic or species restricted to particular locality. We can see the same species here and northern part, and the same species can be found in other countries also. Why
yeah, yeah.
this? Why this particular group of organism doesn't show that type of endemism in the other group of plants or other group of fungi or other group of organisms? Is there any ecology or evolutionary significance regarding that?
No, the biogeographical barriers, they don't come in their way. Wherever the spores are there, they move, and wherever they find a substrate, they germinate on that.
Hmm.
So even there are some species which are bark specific. There are some species which are leaf specific, but there are very few, as you rightly said. They do not show any specificities. So they are found everywhere, and most of the species you find them in US. You do not find them in places where they have not been worked out. The
Hmm.
only thing is they have not been worked out, so are not reported.
Hmm.
Otherwise, they are cosmopolitan in distribution.
Yeah. Thank you, sir. I'll keep in touch with you.
Surely, most welcome.
Thank you, sir. Thank you very much.
So to just add up to Srijit's question, that the preservation is just like fungi or bacteria. We just make a scrap and preserved in glycerol and
No,
dispose.
no, 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 no. They
Okay,
are to be preserved dry.
okay, okay,
They are to be preserved dry. What normally done is every in throughout the world. Match boxes, you have match boxes or have boxes of that type,
okay,
so that they are removable.
okay,
So put a paper strip, slightly stiff paper, well, the drawing sheet type of paper,
okay,
cut it to the size of the match box, have a flap outside so that it can be out and you can move. Because once you shake them, the spores will fall.
okay.
So they are very fragile.
Yeah, yeah.
So they have to be dried, and they can be sun dried
Okay, okay.
or dried at 30 to 40 degrees centigrade. If you want to culture them,
Yeah.
then they need to be dried at low temperature.
Okay.
Yeah, if they otherwise the spores will not be viable at 60 degrees.
Yes, yes, yes. So, and for reviving, then these can be revived in the media again. Once
They
dried
can
at
be
low.
revived. Yes, yes. Spores have been revived even after 60 to 70 years.
Okay, that's good. Thank you, sir. Next question is Sashi Rekha, madam. Please go ahead with your question.
Yeah, good morning, sir. It's nice to hear Myxomycetes. It has always been a mysterious group of organism, fungi, or I always understood it as fungi only. But
But
recently,
fortunately, I am not mysterious.
no, you are not mysterious. But recently, my encounter with this Myxomycetes, I have a very old wooden box which was kept in my balcony, and because of rainwater, it used to get wet. But this time,
Simona,
last
Simona
year,
will come.
Stemonitis had come, and there was this fuligo. I think I had just checked it. It was a yellow kind of a thing, and it was oozing out. So I was wondering what it is. So I clicked it, and then when I checked it on Google Lens, because I didn't know where else to this thing, it showed it as fuligo. But now that wood which is there, it is still there. It is dried. That it has become like a cask. So is it possible for that same thing to regenerate this season, monsoon? Will it come
If you
up again?
keep it, if you keep it outside, and if there are spores on the substratum, definitely something will come out.
I have not disturbed it still like that.
So,
Yeah.
if it is wet, it should not be over wet.
No, it is
should
not
have
wet.
little
Right now, everything is dry. It's such a lot of heat wave going on. Everything is dry now.
Yeah.
So it was inside the balcony. I mean, it was not outside where it is exposed to.
No, it can come.
It can come, no? So
It can.
yeah.
You know,
So
just to add to this, you know, Mrs. Neninga Bremikamp in Netherlands, she was a housewife, and she never went to the field for collections. What she had
okay.
done was, she had the pits, and she would put all dried leaves, twigs, and everything in those pits. And hundreds of species she had described just from those pits.
Oh, you're giving me an idea that I can do it. Maybe I should try
You
that.
can put this mat, or it should have some cellulosic base.
Okay.
Thatched
Stemonitis has,
mat or
yeah.
anything. If it is lying outside, something will come out. Myxomycetes are commonly found on them.
But in that same wood, I had also seen one Daldinia species which had come. Then after I tried
No,
to
Daldinia,
take it,
yes, Daldinia can come.
yeah.
Yeah,
So
Daldinia.
probably there are lots of
If it is the wood
species
is
of fungi in
rotten,
that
then
same wood. Yeah.
Daldinia will come if the wood is rotten.
Yeah. It's a very old box, wooden box.
Yeah,
So
yeah. So
definitely. It should.
I have a complimentary question that is coming to my mind, sir. That are these myxomatoes also host? I mean, substrate specific, as you said that if it is wooden, then stemonitis would have been there like that.
Stemonitis normally comes on this shredded base.
Okay.
So if it is a basket
Yes.
on that, then it normally, even in nature, it occupies such type of crevices.
I've seen it on railway tracks, no, which is abandoned tracks. I have seen stemonitis grown on them.
Yeah, surely.
And is
Yeah.
there any specific season also, sir? Just like mushroom, we found only in
Rain.
rain is
Rainy season. Yeah. That's what I'm expecting to see more of it this time.
Rainy season.
Thank you, man.
Thank you
Thank you so much for
Thank
the
you.
question. Thank you. Jitendra, please go ahead with your question.
Hello.
Yeah.
Hello, sir.
Yeah.
Sir, my question is this Trimor is really helping transportation like Tokyo rail system and metro system.
I could not get you.
Please ask
Hi, sir.
again.
Like Pfizer. Hello, sir. My
Yeah.
question is that if Trimor really help in transportation like Tokyo rail metro system, like transportation.
Okay. Sir, he wants to ask that is Slimemore's kind of morphology kind of thing is really helping to design the. There was an article long back, sir. There was an article that the Japan is designing its road system and railway system based on the slime molds.
Yes, ma'am.
Yes. So he
Yes,
wants
sir.
to ask that. Yeah, it's really asking.
So in fabric printing, some fruiting bodies, their structures and their formation has been used in fabrics, but I am not aware of um, how useful it can be in case of the roads or railway.
Yeah, Jitin, I think the kind of they are maybe trying to use the intelligence of these slime molds because they are effectively making branches. So I think probably that may be correlating with the engineering that, okay, there must be some intelligence in the mushroom kind of division and forming the network based on their efficacy for their efficient nutrition, something like that. So I think that must be there. And
Okay.
thank,
Okay.
thank you so much, sir. Thank you. I think these are all the questions that were there. I think I'll take this opportunity to acknowledge Dr. Sridhar sir and Dr. S.K. Singh sir from Agarkar Institute. Both have joined for today's meeting. Thank you so much, both of you, for joining today's talk. Dr. Shnoy, are you there? Can you give a quick last comments from your side?
Thank you, Professor Lakhanpal ji. Thank you very much. It was a wonderful presentation, and patiently listened to questions of the audience and answered them in a very sweet manner. And I'm sure many of us are inspired by your talk today. I know Myxomycetes is kind of not very much appreciated because it is slightly away from fungi, so, but it is, these are very important group of organisms. We will have Professor Stephenson will be giving a talk in the coming month or so about his experience. Yeah, so we would like to have you for in that.
Yeah, surely I will join.